Hello, strangers. I haven't seen you all in forever. Can you hear me on the mic? Is the mic working? Okay, good, because I can't yell. I have to tell you all, you got the experience through my messages that I sent you. After I recorded the videos for you on Sunday, I got progressively worse. Monday, I pretty much slept all day, no joke. Tuesday, I started to feel a little bit better. By Wednesday, I was human again. But I had that experience where, like, when you get healthy, all of a sudden you're like, oh my gosh, I can do all the things. And so then on Thursday, I was like exhausted again because I did way too much on Wednesday. So I'm glad to be back here with you guys. I apologize if my voice sounds a little bit weird. What I learned this week is that the flu is not always a stomach thing. That's what I always thought the flu was. What the flu can be is like a really terrible sore throat and like exhaustion. Like I said, I slept for like 24 hours straight. What I learned this year is I will always be getting a flu shot every year going forward. I used to put it off a little bit, not anymore. I'm getting my flu shot. So thank you all for coming today. We're going to try to make up some of that lost ground. And remember, I changed the date of the exam. Did we all see that announcement? So we usually take exams on Wednesdays. We're going to take the next exam on Friday. So when we come back after Thanksgiving, we're going to have 2 more classes together to finish content. We'll take the exam that Friday. Then we'll have time the next week to do a little bit of final exam reviewing. All right, so I asked you a little icebreaker question to warm us up as we're getting ready to begin. I asked what you're thankful for. So lots of us thankful for family and friends and for sleep. I do love me some sleep. Love that some of you are thankful for me. That's so sweet. I'm thankful that you all came today. That's good, because like we kind of forgot about this class, right? It just kept not happening. Let's see. Wow, somebody in our class is thankful for Gen Chem. Props to you. If Gen Chem is your thing, man, chemistry was not mine, but somebody's thankful for that. The end of the semester, absolutely. Thanksgiving break, I love it. So apparently we're also very happy that our friends Jackson and Amy are in class today. So props to Jackson and Amy for showing up today. Awesome. All right. Let me spin the wheel actually, since I saw that. This is my wheel for my friends who have not won, theoretically. So hoping I can get a friend or 2 here I haven't got before. Anthony? Anthony here? No Anthony? All right, let's try again. How about Tina? Tina here? All right, Tina, come see me. And I'll give everybody a chance on my big wheel. Jackson, it was almost you. No? Goodness. We're hit and miss today. One last time, then we'll dive in. How about Savannah? Savannah here? All right, Savannah, awesome. By the way, did you all see that ad before it went away? It was a quiz for what squishmallow are you? Did anyone else see that? Like, that touched my soul. My daughter loves squishmallows, so I should have taken that quiz, man. It would have been the sleepy one. I could be the sleepy squishmallow. All right, today we are going to start by talking about the structure of DNA. We're going to talk a little bit about how DNA is copied. I told you all that on Monday I won't make you come to class. I'm going to record videos for you. But I will also let you know I will be on campus at some points on Monday, so I'll make sure to post an announcement. If you want to come by and chat about anything with me, I will be here. So we're going to start this. I will finish this up in videos for you on Monday. And then when we come back from Thanksgiving, we're going to do that last module, Module 13. So just to give you an idea of where we're going with things. So when we talk about DNA, which is the topic of this module and of our next module as well. Oh, I can't talk over people today. My voice can't handle it, sorry. All right, when we talk about DNA, what this stands for is deoxyribonucleic acid. If I break down that name, it tells me the two parts of this molecule. So the first part of the name is deoxyribo. That refers to the kind of sugar that I use to build it. So this is called deoxyribose sugar. Deoxyribose sugar is very similar to what we'll talk about in the next chapter called ribose sugar, which is what I use in RNA. So maybe you've heard of those two things together. When we look at the DNA molecule, it's a double helix that kind of swirls around itself. On the very outside, there's often two lines that we see. That backbone, or those lines that we're seeing, are these sugar molecules. That's where the deoxyribose is. When we think about the kinds of bonds that hold together all these sugar molecules, I want you to underline, highlight, star, phosphodiester. It's a big, long word. Phosphodiester. That's the name that I use to describe a bond between a phosphate and this deoxyribose sugar. So all along the outsides of my DNA double helix, I have a type of bonds called phosphodiester bonds. These are strong bonds that are going to hold these sugar molecules together. In the middle of my DNA molecule, I have what are called nucleotides. So the nucleic acid part of the name of this molecule is where we're talking about the nucleotides now. So when we talk about nucleotides, their job is to store genetic information. So way back in module 2, when we were talking about the types of molecules we have, remember that nucleic acids were molecules that had information. The information in a nucleotide is stored in something called a nitrogenous base. So the nitrogenous base is the part of the nucleotide that gives it that letter that we're used to seeing. So when you think about DNA, you probably think about A, T, G, or C. Those are the nitrogenous bases. Those are the parts, if you look over here, that are here in color. So the nitrogenous base has the different letters to it. All of those, when I talk about a nucleotide though, we're also going to include that sugar that we talked about before. But the actual genetic information, it's in those letters, the nitrogenous bases. There are two kinds of nitrogenous bases that you need to be familiar with. The first kind are called purines. So there are two bases that are purines. That's A and G, adenine and guanine. There are also two bases that we call pyrimidines, and these are C, cytosine, and T, thymine. Now, I like to remember who's who by this little statement here, cut the pie. So cut the pie, cytosine and thymine are pyrimidines. So I put a little pie here since we're coming up on Thanksgiving, right? I put a pie next to thymine. I put a pie next to cytosine. When we look at these bases, these ones only have one carbon ring compared to my two purines that actually have two carbon rings. So when we're taking quizzes or exams, I promise you I'm not going to put up one of these things and be like, which particular base is this? I'm not going to make you memorize their structure. But I might put this up here and say, is this a purine or a pyrimidine? It's really easy to recognize. Pyrimidines have one ring, purines have two rings. So make sure we know who's a purine, who's a pyrimidine, cut the pie, and make sure you can identify them with the pictures, one ring or two rings. Like we normally see DNA, DNA exists in a double strand, meaning it's got two strips of DNA that are next to each other. The fact that it's double-stranded actually makes it really stable. You could actually isolate DNA. I'm a nerd, so I've done this at home. You can isolate DNA from strawberries, and it just like chills out at room temperature. So DNA, very stable. RNA, like we'll talk about next time, not. So double-stranded DNA, the two strands in DNA are complementary. This means that they're mirror images of each other. And after you've written down that they're mirror images of each other, the other thing I want you to write down about them being complementary, I want us to write down in all capital letters, they're not the same. Not the same. When I say two things are complementary, this does not mean they have the same sequence. It means that if I have the sequence of one, I can predict the sequence of the other because they're mirrors of each other, but they are not the same. So in a minute, when I ask you to build me a complementary DNA strand, it's never the same as the one that I gave you. Keep that in mind. And I think we already talked about this before, but when I'm building a complementary strand, every time I have an A in one strand, I put a T in the other, or vice versa. If I had a T in one strand, I'd put an A in the other, and that leaves for me C and G that go together. So we need to know which of these bases, number one, is a purine or a pyrimidine, and number two, who pairs with who. Because I'm promising you on the quiz or the exam, you're going to see some questions like this. So I'm going to stop talking for a moment and give you a chance. I want you to, if you have not already, build me my complementary DNA strand to this strand of DNA that we see right here. So I've given you a few letters. Why don't you build me underneath the complementary DNA strand to this one? So this kind of activity should be pretty easy for us, pretty fun compared to some of the other things I could test you on, right? So when I look at this strand, my first letter is a C. Every time I have a C, I'm going to put a G in my complementary strand. And vice versa. I had a G up there, so I'm going to put a C in this one. Now I bump into an A. Remember that A and T go together. So we work our way through our strand. And this is the sequence that we should get. So we need to know how to use the sequence of one strand to predict the sequence of the other strand. When we look at a DNA strand, since we know which bases pair up with each other, we can make some predictions about the DNA just based on its sequence. So there's this thing called Chargaff's rule. I don't care that you know his name, but I do care that you understand what his rule says. So what his rule says is if I know what percent abundance one of my nucleotides is, I can actually predict the percent abundance for all the other nucleotides as well. So when I have a number, say I give you the percent of adenines, A's, in a DNA molecule, you can directly, very easily, predict the percentage of T's, because those are always going to be the same. Those percentages will be the same. And the same thing goes for G's. If I give you the percentage of guanine, you can very easily predict the percentage of cytosine. But you can also use the percentage of one nucleotide to make a prediction about all of the other nucleotides as well. So I want to work through this problem with you so we can get some practice with this. Here in our practice problem, I told you I've got a molecule of DNA. 15% of my bases are T's. So one of my letters, I've figured it out. Now I need to make predictions about my three other letters. So let's start with the one that's going to be the easiest for me. When I have the T's, a T in one strand, what do I pair T with in the other strand? What goes together? A, right? So thymine and adenine always go together. If I know that this strand has 15% of T's in it, then its complementary strand, the molecule together, is also going to have 15% of A's. Because for every T that's in that DNA molecule, there's a matching A that goes with it. That's my easy prediction. I can do that one right away, no math required. But I also asked you about G and C. Here's where we have to do a little bit of math. I know that 15% of my bases are T's, and I know that 15% of my bases are A's. When I look at how much of my DNA has been used up so far, 15 plus 15 is 30. So 30% of my DNA is spoken for. It's got A's or it's got T's. I also have those G's and C's, though, and they're making up the rest of my DNA molecule. So my total DNA molecule has 100%. I'm going to take away my amount for my T's and A's. What I have left over is 70% of my DNA molecule made out of G and C. And for me to figure out how much is each, remember, these guys are complementary, so the amount that I have left, I split in half. Half of those numbers, or half of those letters, are going to be G's, 35%. Half of those letters are going to be C's, 35%. I want you to find either a space on this slide, or I want you to flip over, find a blank piece of paper. I walked you through this one. Now I'm going to give you a new one that I want you to work on with your neighbors. Let's suppose that I told you we have a DNA molecule that is, trying to figure out how mean I want to be. Let's do 27%. I'm going to do an odd number on you. 27% C. That's the one I told you. Now with your neighbors, let's do some math. 27% C, I got to predict all my other bases based on this amount that I have for C. Let's do some of that math. Thumbs up if we've solved it. How many of us have solved it? Okay, looks like most of us have. So let's check with each other. If I have 27% C, I'm going to have 27% of another base as well. What's the one I'm going to have 27% of as well? G, right? C and G always go together. Okay, so now I'm going to add together 27 plus 27. That gives me 54%. 54% of my genome is taken up with these two, which means that I have 46% of my genome left, and I divide that equally between my two other bases. So 23 and 23 for adenine and thymine. I am promising you on the quiz and the exam, we will see questions like this. So make sure we're comfortable with how we would do these calculations so that when I give you a number, you can plug it in and predict any of them. When we look at a DNA sequence, we can figure out who's a purine, who's a pyrimidine. We can figure out the complementary bases, And we can also go through and actually figure out how many different chemical bonds are holding together that DNA molecule. That's because inside our DNA molecules, our nucleotides connect to each other, so the two halves of the helix connect to each other in very specific ways. So when I talk about the way that an A and a T connect to each other, an adenine and a thymine connect to each other, I use 2 hydrogen bonds to do that. That's the kind of bond that they have. That's different when I start comparing it to G's and C's. When those two connect to each other, they're going to use 3 hydrogen bonds. Hey, so not a trick question. If I'm thinking about which is going to be a stronger connection, do we think there'd be a stronger connection between A and T or G and C? Not a trick question. G and C, right? They're connected in 3 places. So check this out. And again, I'm not going to give you a nucleotide and ask you its name. But check this out. Guanine and cytosine, these 2 are paired together, remember? Guanine has 1, 2, and 3 places where it wants to form a chemical bond. And so does cytosine. 1, 2, and 3 places. They both have 3 places to form bonds, which is why they connect with each other. Adenine and thymine, though, only have 2 places where they want to form bonds. This is why I never pair up wrong nucleotides with each other, because they each have a specific number of hydrogen bonds that they're going to make. I also want to point out for you, remember our pies, right? Cytosine and thymine, my 2 pyrimidines. Something I didn't mention before, but check this out. A pyrimidine always pairs with a purine. And we see that here too. This pyrimidine is pairing with a purine. So it always goes like that. You know, we were predicting our percentages, right, with the different letters. We can actually make the same kinds of predictions with purine and pyrimidines because we know that they always go together. They're complementary to each other. So let's take our DNA sequence building one step further. What we're going to do, first, we're going to build a complementary sequence. Then I want you to tell me how many phosphodiester bonds I have and how many hydrogen bonds I have. Now, it's been a minute since we talked about these. Phosphodiester bonds are in between my nucleotides. So when I connect A to G, that's 1 phosphodiester bond. G to T, there's number 2. So phosphodiester bonds connect each letter to each other. And then remember, I use different numbers of hydrogen bonds for different bases. So let's build the complement. Let's count the bonds. I'm going to start working on my complementary DNA sequence, the really easy part, right? So my first sequence, I've got an A put in a T, G, put in a C, got a couple of A's, G, and T. There's the easy part, getting that sequence in there. Now, I asked you about the phosphodiester bonds. So we talked about where these are. These are between my individual bases. I've got one bond in each of these places. Check this out though. I also have one bond up here too. Went a little tricky on you. This is a double helix, 2 strands. So in my bottom strand, I have 1, 2, 3, 4, 5. In my top strand, I have 5, which means in this DNA double-stranded molecule, I've got 10 phosphodiester bonds. 5 in each half. 5 in each half. Now I'm going to count my hydrogen bonds. And remember that hydrogen bonds have different numbers depending on the letters that I'm connecting. So when I connect an A and a T to each other, remind me, how many bonds between an A and a T? Yeah, that's 2, right? So I've got 2 right here between A and T, and another 2 with my A and T, A and T, A and T. There we go. Anytime I have a G and a C, remember that's going to have 3 bonds. 3 bonds. So the number of hydrogen bonds I have, let's do some quick math, 5, 9, 12, 14. 14 hydrogen bonds. The way I got these numbers, first was looking at where the different types of bonds are. Are they between the letters in the backbone, or are they between the letters across the strands? And then, with those hydrogen bonds, I had to look at the bases. So, use what bases you see to help you with predictions about hydrogen bonds. Use the sequence to help you figure out phosphodiester bonds. They're a little bit easier because we don't have to do any weird math with them. We just count those ones. Up until this point, I haven't really talked with you about the 5' thing and the 3' thing. You've seen it pop up on some of my slides. Now I'm going to tell you what it means and why it's important for us. So when we look at the DNA double helix, we would say that the 2 strands that are connected to each other, we call them anti-parallel. And what anti-parallel means is that their backbones, their sugar phosphates, are oriented in opposite directions. So one of them, the 5' end is up, and it goes down to the 3' end. The other one, the 3' end is up, and we got the 5' end down below. That's what anti-parallel means. When we talk about these numbers though, 5', 3', why are we just making stuff up? Well, it comes down to chemistry. We need to make sure that we know what we find at each of these ends of a DNA strand. So when I'm looking at the 5' end of a DNA strand, I'm looking at the sugar phosphate that has a phosphate group hanging off the end. So my deoxyribonucleic acid that's got the phosphate sticking up, that's my 5' end. The end that has a hydroxyl group, an OH, that's going to be my 3' end. So we need to make sure, as we're studying the structure of DNA, we know that the 2 strands together, they're opposite, 5 to 3, 3 to 5, but we also need to know where those numbers come from. 5' comes from having a phosphate group hanging out there in the 5th position. 3' comes from having a hydroxyl group hanging out in that 3' position. And this matters because as we start talking about copying DNA or building a new DNA strand, we can only add things at the 3' end. And once you write 3', I need you to underline, highlight, star that. I can only add things at the 3' end. It's because of my chemistry. So remember, 5' has a phosphate, 3' has a hydroxyl. Having different functional groups is going to make it impossible for me to add DNA nucleotides at one end, and the way I do it at the other end. So if I'm trying to build a new strand of DNA, I'm trying to copy something, make it longer, what I do is at that 3' end, I'm going to steal a hydrogen from that hydroxyl group we were talking about. So remember, hydroxyl group is an OH. I'm going to steal that H from my OH at the 3' end. That gets us ready to attach a new base. Then I'm going to bring in that new base that I'm adding, and I'm going to take away from that an OH group. So what I'm bringing in has that phosphate. When phosphate is by itself in the wild, it actually is phosphoric acid. So I steal OH from it. You can see it right here. I'm going to steal the OH. I'm going to steal this H. When I steal those things from each other, I build that bond called phosphodiester bonds. So why do we care about 5' to 3'? Because it gives me an idea, where am I going to be able to attach new nucleotides when I'm trying to copy this molecule? I have to attach them to the side that has the hydroxyl group. I have to attach to the 3' side, because the new base that's coming in is coming in with its phosphate group. The phosphate group there at that 5' side. So remember, I've said it multiple times, we'll never forget it now, right? Phosphodiester bonds connecting together the backbone, putting all my DNA nucleotides together. Remind me again, which kind of bond held together my two halves? What holds the letters together in the middle? Hydrogen bonds, yep. Hydrogen bonds hold the halves together. Phosphodiester bonds hold the backbone together. We're going to start talking about DNA replication today and talk about it a little bit more in those videos next week. One of the big ideas that I want you to know now about DNA replication is that when we do DNA replication, it's something that we call semi-conservative. So I'm going to walk through the process of DNA replication with you to help us answer our question of what makes it semi-conservative. When I'm ready to copy DNA, I have a DNA molecule that's double-stranded. I've got my double helix thing going on here. I can't copy DNA when it's stuck together with the hydrogen bonds. So the first thing I have to do is I have to open up those hydrogen bonds. I have to split apart my two halves of the double helix. When those two halves are open, then enzymes that are inside my cell will come through and build a complementary DNA strand to each of my two halves. Each of my two halves serves as a template to make a new DNA strand. By the time I'm done with that template strand, I end up with two new molecules of DNA that are half old and half new. So calling it semi-conservative, semi means partially, conservative means the same. Semi-conservative replication means half of my DNA is old, half of it is new. So I've got a template strand and a new strand. That's what makes up my ultimate new strand of DNA. So I know the word semi-conservative looks really crazy. Semi-conservative replication, all it means, half old, half new. That's the replication process. To do replication, we use a few particular structures. So the first structure we're going to use is something called the origin of replication. So the origin of replication is where copying is going to begin. That's what this word origin means. It means the beginnings. My origin of replication is a particular place in my DNA where that DNA can open up and create a space for me to start copying it. When I open up my double-stranded DNA, I make something very technically called a replication bubble. Who says we can't have fun in biology, right? We're making a bubble. The replication bubble So I open up the origin of replication. I've got this bubble-looking space where I can then go through and start to build new DNA inside of. But when I just open up one part of my bubble, I've got a whole lot of other DNA that I've got to get to. So that other DNA that I'm going to get to, I have to expand my bubble, going 2 ways to get to the rest of that DNA. To help me expand my bubble, I see these things called replication forks. So replication forks are like what you'd call a fork in the road, where the road splits 2 directions. That's where my DNA splits into 2 directions. So we first open up the helix at the origin of replication, we make a bubble, and then those replication forks move away from each other, allowing me to open up the entire DNA strand. As I open that whole DNA strand, I can copy or build a copy of the information that's there. Now, when we think about copying the human genome, which is really big with all of our 46 chromosomes, we're not just going to start with one origin of replication on a chromosome and let it go. Because if I started in one place, I have literally millions of nucleotides on each chromosome. It's going to take me forever. So on human chromosomes, we'd look at a piece of DNA and notice that this piece of DNA has multiple different origins of replication, all these little black spots. So I open up each of those around the same time, when it's time for my cell to copy its genetic information. As I open them up, I make a new bubble. And on the 2 edges of the bubble, I've got those forks. Well, those forks start moving away from each other. They're moving in those opposite directions. But at the same time that this fork is moving to the left, this one over here is moving to the right. So we end up with forks meeting each other. And when they meet each other, I just completely separate the 2 halves of my DNA double helix. So in our chromosomes, when we copy them, we do small-scale copying right at that replication bubble stage. But as we zoom out and look big picture, the way we get all of your bases copied into a single piece of DNA, like we see here, is all of those bubbles meeting and us stitching together all of those pieces. So this is an overview of how the process of replication works. We open at the origin, make a bubble, bubble expands with the forks, and that's how we're going to copy things. When I post some videos for you, I'm going to go into a little more detail. I'm going to tell you things like, what's the name of the enzyme that opens up the double helix? What's the name of the enzyme that adds nucleotides? What's the name of the enzyme that stitches things together? So when we start learning about those specific enzymes, always come back to this framework, thinking big picture. I open up the DNA and I copy it in 2 different directions. We will see, though, that we make one of our new strands of DNA continuously. I can add bases to that one all the way. The other one I'm going to make in little choppy pieces. That comes back to what we talked about with building DNA. Can you remind me, is it the 3' end or the 5' end of a DNA strand that I can add new bases to? Which side? Yeah, 3'. So when I'm building a new strand of DNA, I'm hoping that the side that I'm trying to build on is 3'. But some of my DNA that I'm copying, it's actually not. So we have to do a little bit of backwards working on that one. On that note, I am done with what I wanted to cover with you today. I will post you an announcement with the links for all of the videos that will wrap this up. And I'll also let you know when you could stop by my office if you have questions. Have a great Thanksgiving break. I will see you all in a couple of weeks. Oh, here, I owe a couple spinners. Let me do a couple quick spinners. Marissa? Marissa here? All right, Marissa. Let's try to get another friend. It is slim pickings on the Play-Doh, but I've got lots of skeletons. And let me dig my stickers out of my bag as well.
I just have a quick question. So I'm looking at the exam schedule, and I don't see the final exam on there. I'm just curious what day
The final exam is on Friday, and it is at our normal class time. So 11 o'clock on Friday. So it's the Friday after we're taking the regular exam.
Okay.
It's the Friday of finals week. Feel free if you want, find a couple you want.
Okay, so the 9th, I believe. Yeah, because we're not here by the 6th.
Correct.
Okay.
Yes. Yeah. The 9th.
Thank you.
You're welcome.